So let's get started. This session is going to be around managing tourism and other human impacts on protected areas. And our first presenter is Nima, and he's going to be talking. Look what happens when you get older. I don't have my glasses on. I have to do that to actually see what you can. I'm going to let you tell us.
No worries. Thank you very much.
Go ahead. You've got a 5-minute talk.
Sounds good. Yeah, thanks everyone for coming back so quickly from lunch. I know we're running a bit behind schedule, so I'll just get started. I'm Nima. I'm a master's student in computer science from the Hamburg University of Applied Sciences in Germany. And my research group and I, together with Sandparks, have been working on a model for analyzing traffic flow in the KNP, which I'd like to tell you a little bit about today. The goal of the model is to provide decision support to Sandparks with respect to visitor and traffic management. And it's still under development, but we hope to deliver the finalized model within a couple of months to Sandparks. So before getting into the model, let's just talk briefly about why this kind of analysis might be interesting. For one, visitors are an important contributor to Sandparks' revenue, but of course there are also sustainability-related concerns when looking at traffic and the amount of traffic that goes through national parks in general. So a question with respect to the visitors is how to improve the visitor experience. And in this case, how to reduce the number of traffic jams that occur in national parks. And there are actually quite a few ideas and approaches to tackling this issue, but the problem with testing these in the field is that they either tend to be expensive or might have negative effects or ones that we just didn't expect to happen. And usually because of the complexity of these traffic systems, the effects, whether good or bad, tend to take a long time to actually reveal themselves to us. So we think, as computer scientists, a good approach in this case is to design a model to enable management and stakeholders to run their initiatives in a simulation and to see their effects there as opposed to directly in the field. So this is an agent-based model. And without going into a lengthy definition of what an agent-based model is, I'll just say that we need two things. We need an environment, and we need some agents. For the environment, we actually were provided with quite a few georeferenced data sets directly from Sandparks, which we integrate into the model to give our agents an environment to move around in. And as you can see in the figure here, this mainly consists of a road network that the agents can move around on, and also several points of interest, we call them in general, such as the KNP gates, rest camps, picnic spots, and so on. For the agents, we distinguish between three main types. So the commuter at the top might be someone who works in the KNP but lives outside of the KNP. So that's a person who maybe commutes into the KNP on a daily basis and heads to a specific destination. Then for the visitors, we have daytime and overnight visitors. Here actually on the right, this little icon-based diagram shows a typical routine of a daytime visitor. So that consists of entering the KNP through a gate, then doing some random exploration in search of wildlife for some time, probably stopping either to go to the restroom or to have lunch, or maybe both, at a rest camp, picnic spot, or something similar, and then probably repeating that cycle, but making sure to leave the park through a gate again before closing hours. So that's how we model, for example, the daily routine of a daytime visitor. And of course, we also have OSV drivers, and there we distinguish between the camp-based ones and the external ones who come into the park to do their tours. And finally, of course, we also need to have wildlife sightings in the model for visitors and OSV drivers to interact with if they can spot them. Those are events that are positioned randomly by the model along the roadside and stay there temporarily. And this is just a quick visual to give you an idea of what happens during simulation. Down here, we see two visitors. They entered through Malolane Gate and are exploring randomly. And up here are two commuters who came in through Fabeni Gate and are heading straight to Skukuza Camp, presumably because that's their location of work or their place of business for that day. But of course, we want to run the model with more agents. These are heat maps that were generated with 100 visitors. So first, over here, we see a traffic density heat map, and in general, the darker the square, the more traffic occurred on that underlying road section under the square. In the middle, we see a map that shows the location and intensity of traffic jams. Again, for example, up here or here were traffic jams that took quite a while. And finally, here we see the location and duration of sightings where people actually stopped to look at wildlife. And we hope that with these kinds of outputs, among others, we can assist sandparks by running simulations and testing their initiatives in a model, again, as opposed to in the field. And that's all I have. Thank you very much.
Thanks so much, and very good time management there. Thank you very much.
Thank you.
Super interesting. Those little simulations reminded me of my Mapani worms. Oh, I said I wasn't going to mention them. Thank you.
Thanks.
No, it's fine. Okay. We're ready for you. So Claudine's going to be talking about best practice principles for waste management in South African protected areas.
I just need the remote.
The remote I have. Here we go.
Thanks. Thank you very much. Okay, so it's a quick 5-minute presentation looking at proposing best practice principles for waste management in protected areas. And my co-authors are sitting there at the back, the policy guys from Monday. So waste management has been identified as one of the biggest challenges, or one of the most significant challenges that we have in and around protected areas. And we have realized that we need some sort of a standardization of waste management practices in protected areas. So this paper basically aims to very quickly propose best practice principles towards waste management in protected areas. Okay, so our method, how did we go about proposing these best practice principles? Firstly, we started with Section 2 of the National Environmental Management Act, or NEMA. Sam, you mentioned the duty of care earlier today, so that is the duty of care, the proximity principle, cradle to grave principle. So that was basically our point of departure, and then we contextualized those principles in line with other waste-related legislation like the National Environmental Management Waste Act, the Waste Management Strategy, Protected Areas Act, Biodiversity Act, and Strategy as well. Okay, after we had our basics in place, our basic principles in place, we also wanted to contextualize or align these principles with international best practice. So we did a literature search and then contextualize our principles based on waste-related principles or guidelines internationally from the US EPA, European Union, USAID, the World Bank, etc. And we also, apart from waste principles, looked at, are you not seeing what I'm seeing? Okay, we like steps behind. The protected areas principles were also brought into consideration. So there we looked at the IUCN guidelines for ecotourism, for visitor management. We also considered the International Association for Impact Assessment and their principles for biodiversity impact assessment, the USAID guidelines on ecotourism, and then also World Bank guidelines for sustainable tourism. Okay, I think we're not aligned in terms of where I am and in terms of what you are seeing. Okay, let me continue to just save time. So the third step of our process was then to take these principles and to workshop them with a specific set of role players. And then we came up with 10. Sorry, we're jumping around now on this, so I can't really see where I am. Yeah, so we came up with 10 key principles, and these were principles considering how we need to manage waste, not only inside protected areas, but also to minimize the impact of waste outside of protected areas. So our first principle was looking at the disturbance of ecosystems and biodiversity, so managing waste in such a way that we minimize disturbance of ecosystems and irreversible loss to biodiversity. The second principle, let me just see if we can get on the same page. Sorry. The second principle looks at pollution that needs to be avoided, minimized, or remedied, looking at our pollution control hierarchy. Principle 3 looks at the implementation of the waste management hierarchy. And now it's a problem. I can't remember the rest by heart. So if you can just skip to the slide where we have principle 4 onwards, please.
Pause for just a sec.
Perfect.
Let's just see if we can just get the two to speak to each other. Just bear with us, please. Don't you have anything in the bag you can do for us, like a little dance or...
Singing.
Just to keep everyone busy, yeah. Singing would be great.
No, I don't have the best singing voice. Let's just hang in there. Okay, there we are. Let's see if we can see it on the big screen as well.
Pausing just for a sec.
There we are. So there we see the first principles that I spoke about. The fourth principle is all about effective governance and ensuring effective governance, the coordination of waste management services, and also providing adequate waste management infrastructure. Principle 5 looks at the sustainable use of resources, promoting the sustainable use of resources, but also viewing waste as a resource. 6 looks at avoiding and managing negative impact on the environment, but also on environmental rights of people. Partnerships and participation, we spoke about that this morning as well. Relationships, very important. So fostering the building of partnerships and relationships and participation. When we look at waste management, looking at community well-being, empowerment through and contribution to livelihoods. Skills and capacity building through education, awareness, and knowledge sharing, and then also looking at affordable and financially sustainable waste management measures with the potential also of cost recovery of whatever the service or the infrastructure is that we are using. So in conclusion, we believe that these principles provide a valuable basis for evaluating your current waste management practices against to see how are we doing, are we in line with best practice, and then also adopting of these principles or alignment of our current implementation measures, our management strategies and plans, our standard operating procedures, and also including these in environmental impact assessments for protected areas. And that is it. Thank you. Monty.
Great. Thanks so much, Claudine, and thanks for being so calm with our little technical glitches. I'm sure we're going to get it sorted out now for the next session. So let's just see how we're doing. We're going to have a look at Montague's, but I think, should we continue with Jason in the meantime? Should we do that? Okay. So that'll be a temporary end of the tourism session. We're going to have Montague just now still. But we're going to now start with the disease ecology session. My forte, as you know everything about disease ecology. That's why they chose me for this session. But there were a couple of announcements that I was supposed to give you. The first is about the bus for this afternoon. So there's a game drive. The bus is going to be leaving from here just before 6. So after this session, there's a cheese and wine poster session, and then the bus will be leaving just before 6. So don't be late. Remember, you need your indemnity form that has to be signed. And then you'll go on a little night drive, and then you'll be delivered to dinner. and then you'll come back after dinner. And then you just are going to need, anyone who's actually leaving the park, you need an exit permit. So they're not going to let you out if you haven't got an exit permit. So you need your entrance permit to get an exit permit. And you can also get that from Jackie. Okay, so we're going to start with the disease ecology session. Jason Donaldson's going to be talking about fire and macroparasites for 15 minutes. Thanks, Jason.
Thanks, Lou.
Now you've got to wing it if you don't get your slides.
Yeah, it's going to be tough. Hi, everyone. Thanks for joining me this afternoon. And thanks for giving me the big room and disease ecology, the big room. So a lot of people have asked me, why am I in the disease ecology section? It's kind of unusual for me. And really the thing is, I couldn't move and do anything in this space if it wasn't for this great team that we're operating with up in Tanzania. It's a really fun and collaboration between a lot of people, and that's allowed me to kind of shift some of my ideas into this parasite dynamic space. And so hopefully it's as fun for you as it was for me to kind of shift and start thinking of a lot of these ideas in a different space. So I'm going to start with really what's a very basic Savanna start, which is that fire burns in savannas, burns quite a lot. And there's herbivores in savannas, wild herbivores, in huge numbers, we know this. And also there's livestock, again, really big numbers of livestock, very important for human livelihoods. So savannas play these really key roles in human livelihoods and in animal welfare. And so the question then is, what happens when these two things interact? And this is the space that I've been working in a lot in the last few years. And we have very good understanding of what happens when you burn in savanna landscapes with herbivores. The grass greens up after a fire. All these herbivores pull into these really fresh growth areas, and they start grazing very intensively in those areas in what's been called pyroherbivory. And this has been really demonstrated all over the world in a lot of different systems. And this is just a really clear example of where you've had burn and this nice fresh growth and your tall grass areas here. So there's nutritional benefits, and then for wild herbivores, there's also your anti-predator benefits. So we know a lot about the feedbacks between herbivores keeping these grasses short after fire, but we do not have a very good understanding of what that means for macroparasites, and especially environmentally transmitted macroparasites. So the question we're kind of addressing in this talk, or that I'm going to try and address in this talk, is what's happening in these two spaces. So you have this burnt area that's flushed green, and you have this this unburnt area on the right here. What does this mean for macroparasites that are transmitted environmentally? You would expect that with fire, because these environmentally transmitted parasites have life stages that are outside of herbivores that they need to go through, they would be kind of exposed to fire effects. So there has been some work on ticks within Africa. In African savannas, Winston Traub's done quite a lot of work on this, showing that tick numbers can decrease, but there's really not been a hell of a lot else done on parasites and environmentally transmitted parasites. So really, how does this landscape affect these herbivores that can take this up is the overall question here. So if we look at that, we have this question, what do fires in savannas do to macroparasite distributions? And we split it down into two groups. So what is the immediate effect of fire? And then after that, what is the effect of pyroherbivory, that response of herbivores to fire? And so I'm focusing on gastrointestinal nematodes. So just to give you an idea of this life history, you have these adult adult nematodes that live within herbivore hosts. They then lay eggs in the gut, which come out in the dung. The eggs then mature in the dung, and they hatch into larvae, which climb onto the grass layer and then are eaten by the hosts. Okay, so that's the system I'm focusing on here. And as you can see, this section here is really important for us in terms of what's happening with these parasites when they're out in the environment. That's where fire and herbivore effects can really take place. And so if we imagine a system where you've got some grass layer, you've got a base level of parasites within that environment, and then you have a herbivore population that has some infection rate, right? This infection rate isn't representing an individual that's infected, it's representing like a proportion or a parasite burden within the population. So these parasites are really small. they're out there in the grass layer, and they're exposed to the environmental effects, right? They're tiny, so you can imagine desiccation, heat, all of those things can kill them. So obviously, what you would expect is that when you light a fire and it burns through this grass layer, you'll kill all of these parasites in the environment. So that was our first hypothesis, was that these fires can kill the larvae in the environment. But there's a second hypothesis here that could potentially occur, and this comes from, there's some evidence that these nematode larvae dig down into the soil, and Tertia gave this great stat yesterday on the fires down in the basalts, that a lot of those fires were 500 degrees Celsius plus, but within the soil there, below ground, it was only up to 34 degrees Celsius. So you can imagine for a larvae, if it manages to get in the soil, it may have a real chance of surviving these fire events. So that was the alternative hypothesis. So what we did to test this is we had two exclosed plots. We added dung to them. So day 0, we took a sample of the larvae. We added dung to them. Day 14, we assessed the larvae again. Then we burnt those plots, and then we looked, when it greened up, what the larvae were doing. And these two great guys, Aiden Trentinus and Basil Senzo, were the ones doing all the work on this in the lab. And so, yeah, I owe a lot to them. Most of the data presented in this has been processed by them in the lab. So this is what we found. It's pretty straightforward. So this is larvae on the y-axis, the days on the x-axis here. And so we added this dung. You see the spike in larvae goes up really high. And then we burn, and you see this comes all the way down to 0, and the ones in the grass, which didn't burn, still survive. And so fire kills all of them. You might ask, okay, what's happening in the soil there? Well, we found one larvae in the whole experiment in the soil there, which made a really boring graph, because it just went from 1 to 0. So it looks like they're not spending a lot of time in the soil, and fire is killing them in the environment. So that leads us to this step. So we've got fire, it kills the larvae. And we want to see, okay, well, do the herbivores in the Serengeti ecosystem, are they responding to these pyric events, these fires, okay? And so quickly, we had these 43 we had these 43 by 20 meter plots that we set up in burns that already occurred. We didn't want to set our own fires, so we went to where Tanapa, the Tanzania National Parks, had burnt, and we set up these plots all over there. And we paired them burnt and unburnt, and we looked at, we ran dung transects through them, and we monitored them every month for dung just to see the response of herbivores. And so what you see here is on the y-axis, you've got dung piles per meter squared, and then we've just got a timeline here. The fires all occurred in this July-August period. And so what you see is initially, there's no response in September. A little bit of rainfall, though, and then you get this response. So on the fires, we're getting higher dung piles, which is high inputs of eggs into these fires, burnt patches compared to the unburned patches. So we have this pyric herbivory effect. We have this response of herbivores to the fires. So the next step in this is, okay, well, if we have that, then you have shortgrass patches that will occur there. You add a lot of dung into these spots compared to the unburned patches, we should see an increase in L3. That was our expectation. So to test that, we went back to these plots, we walked these W transects, we did 2 of those, and we sampled pasture, took that back to the lab, and we looked at larval counts within the pasture loads. So what does that come out as? Again, on the y-axis here, we've got L3, so these are the larvae that we are in that grass layer per kilogram of grass biomass, and we've got these months of the year. And so the initial thing that we see is straight after the fires, there's nothing, as we expected from our experiment, there's nothing in these burnt patches. There's no larvae, much higher in the unburnt patches. But it recovers really quickly. So that dung is seemingly having an effect. But what was really interesting and quite confusing is if we look in November, the dung was the highest in October. So you'd expect that following month to have really high L3 densities. And you don't. You see much lower in the November plots. And so what I want to highlight here is that in October, there was almost no rainfall. And there's no temperature on this axis. Sorry, this rainfall is from here. There's no temperature in here, but it was really hot in October as well. So that got us thinking, okay, maybe there's an environmental effect here linked to this grass layer. And so there wasn't a very strong dung effect, and we became a bit more interested in this reciprocal or indirect effect. The grazers are grazing this grass short. Is there a microenvironment or microclimate effect that is affecting these larvae? Okay, so the first thing we did is we put up these sensors in the ground, which had a little thermocouple measuring temperature at the ground level, and then we had those coupled with weather stations, and we measured grass biomass, through this experiment. And what you can see here is on the y-axis, this is the difference in the maximum temperature. So the maximum temperature in a day, what is it at the ground level, what is it below the grass layer? And then standing grass biomass. And very simply, as you increase your grass biomass, you decrease that difference in maximum temperature. So 0 would mean it's the same as the maximum in ambient, okay? Same as the maximum at the weather stations. Which means you could, if it's a 30-degree day up here, you could be experiencing 60 degrees Celsius temperatures in the short grass patches. And when we compare that to the change in larvae within the environment, so this is month-on-month changes, so how are the larvae responding 1 month to another? What you see is the daily ground temperature, the mean daily ground temperature, the temperature from those sensors, is that you get this decline. So anything below 0 means that you're actually losing larvae in the environment, and anything above this line means that you're gaining larvae in the environment. And so you see there's this temperature effect at the ground level. So the hot ground is killing these larvae in these short grass areas. The other response that we found is that grass leaf moisture content mattered. So if you have wetter grass, so again, change in larvae per kilogram of grass biomass. If you have wetter grass, you have less mortality in that in the larvae and can be increasing in the larvae. And the reason I put this dung piles per meter squared here is just to show you there's no dung relationship. So this did not, change in larvae had no response to dung inputs. So it's really being driven by these environmental effects. And so what we see is we've got pretty good support now that you increase the number of herbivores, they keep the grass short after this fire, that affects this microclimate, which reduces the larvae in the grass. And then the next step in this would be, okay, well, does the larvae in the grass actually, the changes in these larvae along here, along this bottom axis here, do they actually affect the population level infection rates? So that's what we're looking at here. So what we did is we collected samples of dung all across the system from 5 different herbivore species. This is just an example of topee. So we collected dung throughout the whole year. Each year we collected 20 samples per species. And these are the fires that were occurring during that time. And all I did is I went and looked at, okay, we select a point, what's the burn area within a 5-kilometer radius? And can we get a high burn score or a low burn score? And those were the 2 categories I split these up into. And then we counted the number of eggs in the feces of these samples so that we had a good idea of, okay, what's the parasite burden within these animals given the different sampling of the fire in the system. So I've just got 2 of the species here. It's quite complicated to show the whole figure, but this is topee and wildebeest. And this is strongyle eggs per gram of dung. So the higher this is, the more infected the animals effectively are. And again, just through time here. So we have some relationship here. It looks like down in November, after that hot, dry October, there was much lower parasite loads within animals that were feeding in the burn sites. And again, in wildebeest during that December, January period. So there seems to be some evidence of this, but it's not 100% clear. So although I think there's support for this idea, I think I need to do a far better analysis of this to get a clearer sense. So for now, I'm just going to box this in this broken line, which means I think there's support there, but I would like to do a better test of that as we go. And then finally, to end off here, we've got this system now where you have a fire that initiates these feedbacks with herbivores to keep the grass short. That impacts then the larval numbers in the grass layer, which potentially impacts these herbivores up here. And what I think longer term is going to become really interesting is, okay, if we have these feedbacks established here, how long does this effect last within the system? So when do the animals actually start to experience higher larval loads within those fires? Is there a point where the grass gets enough biomass that it protects these, and then that becomes a cost to the animals, and they decide to move off? And can this help explain why animals are moving off burns through the year and the timing of their movements off of fires during the season? And so, yeah, I just want to thank Tanzanian National Parks and the Tanzanian Wildlife Institute for their support with this work, and everyone for coming. Thank you.
Thanks, Jason. Really, really interesting work. So one quick question for Jason, if anyone has, while Montague gets ready. There we have Alex.
Yes, thank you. Livestock you use sometimes to remove ticks from an area in order to, to leave wildlife without ticks, for example. Do you know in mixed systems, in that ecosystem, do you know in mixed systems if you can have some synergies between livestock rearing, livestock farming, and wildlife in terms of dynamics of pathogens?
So you mean similarities between livestock and wildlife, is that?
Yeah, and maybe some livestock just removing the parasites from the area given their density, and maybe lowering the load in wildlife.
Yeah, so that'd be really interesting. I think we're looking at that with some parts of this project, but in this area, this is central Serengeti, so it's just wildlife that we're looking at. But what our experiments have shown in another system, or another set of experiments, is that you can get that removal effect, so that trophic effect. So you would expect that if you did have livestock that were treated and were coming into the system, not bringing in novel parasites, but removing them, that would be the case. And equally the other way around, I think.
Okay, thanks so much, Jason. So there were 2 more questions, but I'm going to ask you just to hold those until afterwards. We're going to move on to Montague, who's going to give us his presentation on the integrated digital ecosystem for participation in conservation activities. Thanks, Montague. Sorry about the technical issues earlier. We look forward to your talk.
Thank you, Louise, and sorry everyone for the inconvenience. So my topic for today is towards an integrated digital ecosystem for participation in conservation activities, including environmental footprint offsetting. I will be briefly introducing our project. I will look at some of our phase 1 results, and then I will also say where we are right at the moment and look at a few of the next steps. Climate change has a considerable impact on livelihoods in low-income households bordering the Greater Limpopo Transfrontier Conservation Area. The Kruger Park wants to be a catalyst for climate change mitigation, responsible tourism, and rural development, but the Kruger Park has limited resources to fund these strategic aims. So therefore, innovative approaches are needed to pursue and to fund these aims. So against this background, we started off researching, developing, and testing a responsible tourism package with benefits to rural communities. One of the first things we thought we want to do is it should practically be possible for a tourist to offset their carbon equivalent footprint with projects benefiting local communities in the vicinity of the park. When we started the program, from the start, we accept that responsible tourism is not the only important cause to pursue. We also say that responsible tourism is more than environmental offsetting. We acknowledge that, and we also are aware that environmental offsetting has its believers and its skeptics. And unfortunately, accurate measurement in offsetting, not unfortunately, accurate measurement is complex, but unfortunately, some programs use methodologies and standards that are flawed. And we know, I'll say something about that in the presentation. And then we also are aware that adaptation should in some instances be preferred above mitigation, specifically in these contexts. But our endeavor is to grow in our understanding of these complex questions and to research and develop viable ways for tourists to mitigate their footprint with tangible benefits to local communities. So the phase 1 results, we developed a cook stove. It took us about 8 years, but co-creating it with households, but we believe it's well integrated with the live world of end users, and we implemented it in about 6,000 households. We registered it for the gold standard for the global goals, which is a fairly good standard, and we successfully achieved the savings. Some of you may remember, those of you who attended the 2019 conference, we also probed scientific tourists' views on responsible tourism. Obviously, scientific tourism is a very specific type of tourist and definitely not representative of all kinds of tourists, but we were interested to know how scientists think about offsetting. And what was interesting is that we found that 80% of the respondents that completed the questionnaire were willing to pay slightly more for their visit to the Kruger Park if they can be assured that the compensation will go for their ecological footprint. And 83% will be more motivated to offset their footprint if credits are sourced locally. We also built a mobile app and we did our first transaction successfully. Thus, in principle, it's not that difficult to generate benefits locally, to get it. There's some willingness to compensate from tourists and to do it on a mobile app. All of that is possible. However, we are still struggling with a number of challenges. So the first challenge that we're struggling with is that there are quite a number of what we believe substandard programs that are also aiming at reducing greenhouse gas emissions that compete with and undermine trust in higher quality programs. And I'll say something more about that. The other elephant in the room that we all know about is that the bulk of the carbon footprint is actually the footprint traveling to the park. I'll also show you a little bit how that compares with responsibility in the park. And then communicating and information and building trust is quite complex. So looking at the substandard carbon programs, so these programs make naive assumptions about the life world of end users. So you may find that people implement on large scale a one-size-fits-all type of solution. The problem is not always the fact that they are implementing, but the fact that they are not measuring all the correct outcomes. Just to give one example, in rocket stove programs we've been testing, we've seen that if you replace an open fire, which is the baseline scenario outside, with a portable rocket stove, you sometimes find that households actually take those portable stoves indoors, which creates a serious indoor air quality problem, obviously, for that household. So the household, from a health perspective, is in those instances in a worse case than in the baseline scenario. But those types of projects that are only fixated on measuring carbon will not be looking at the health impacts and will not always be monitoring these effects. So this is just one example. There's many other examples. Then some of these programs also do not apply best practice methods. They will, for example, use an unrealistic fraction of non-renewable biomass values. I've seen projects that even use 99% and 100% non-renewable values, which is not really the real state of affairs. They also sometimes work with elevated baselines. Some of these methodologies allows these baselines, and then ultimately there's exaggerated usage of these stoves. They claim that the stove has, for example, if it's a cook stove program, has replaced the open fire, but it has not in all cases been the case fully. So the result is that these programs claim more greenhouse gas savings than is actually happening, and it undermines trust. And I believe that trust in this market, like in many others, is the most important asset that we have. And a credible entity like the Kruger Park that is in conservation for the long run, it's so important that the park make the right decisions in terms of choosing the right programs to work with and measuring these outcomes in the correct best practice manners. And then, just to show that Isaac yesterday said something about the media and perceptions. This is an article that recently was published in the German weekly Die Zeit and also in The Guardian, saying that 90% of rainforest carbon offsets are worthless. And so this message gets into the media. We also did recent, quite comprehensive studies recently. In 2022, NOVA did... We're looking for interventions to reduce domestic wood use, and we did it in a couple of hundred of households. We divided the households in groups of 20, and we also looked at waste burning solutions. So in terms of rocket stoves, we tested 100 stoves in 100 households in 4 communities in 5 groups of 20. So you will see here that it was different types of communities, rural villages with electricity and also without electricity. But what is interesting from the results we received 93 usable results. And from those results, there were only 2 households that used stoves on a daily basis. And the reason we know that is we did very comprehensive measurements. We did eye button temperature sensor measurements, wood weighing, and qualitative interviews. So just to say why we are doing all these things, so with a temperature sensor, you can see physically when people actually made the fire. Wood weighing is still important because what happens is in some households, people may use both the open fire and the new technology because now you can cook 2 dishes at the same time. So you should not assume that if you introduce technology to a household, they will replace the open fire with a new technology. So you also have to do those measurements, properly. So you'll also see that in one community where there was no electricity, a rural community, there was some frequent and occasional use. But in another community, in this one group, we also introduced LPG technology. And in that particular instance, it was a no electricity community. 19 out of the 20 households never used the stove or hardly ever used the stoves. This is just to show you how the eye button measurement results look. So this is 19 households. Each block represents a household. And we did the temperature measurements for a whole month, for the month of August. This zigzag fluctuation is the fluctuation in the daily ambient temperature. And when people use a stove, you can see the spike in temperature. So you can see in this group of 19 households, there were 3 households that basically regularly used the stoves that was introduced. Okay, so that was the substandard programs. The other challenge that we are facing is that the bulk of the carbon footprint, if you just think of a climate perspective, is actually the footprint of traveling to the park. So a typical flight from Germany would be 2.5 tons, 2.7 tons about. And there you can motivate responsible tourism in the park, ask people to switch off lights, drink less coffee, switch off the aircon, drive less, but you have to keep in mind that to get to 2.7 tons, you can have a 25-watt bulb on for 100,000 hours. You can drink 62,500 cups of coffee, leave your aircon on for almost 1,000 hours, have 214 braais, and drive almost 12,000 kilometers in the park. So should we then say that international visits to the park should be discouraged? That's definitely not my view. My own personal view is that virtuality can never replace the experience of a physical visit to the park. You are all here and you will all, I'm sure, agree with me. So therefore, the visiting the park should be a window of opportunity to be converted to a lifelong interest in conservation and a growing sense of environmental responsibility. So if one has that perspective, visiting to the park should be encouraged. And I also think there's no need to necessarily criminalize consumption. We all have to consume. We can avoid and reduce consumption where possible, but there's also some cases where it cannot be avoided. And in cases like that, it makes sense to also have the opportunity to do an offset if you choose to do so. So challenges in terms of tourists, I'll have to be quite quick, is articulating the meaning of offsetting to a diverse spectrum of tourists, distinguishing between these substandard programs and higher quality. It's very difficult for a non-expert to make an evaluation of what is a good, because all projects claim that they are improving quality of life, etc. But we have to find ways to actually communicate that information. And then building trust by transparently showing how offsetting benefits community and then providing an easy, user-friendly way to compensate. I'm now moving to the last part. So what are our next steps and what do we want to do next? So digital networks enable stakeholders to interact and transfer both information and value digitally in a trusted way. The evolution of digital networks create new opportunities. So the internet was actually started in the 1960s. It developed further in the 1980s, and then when the World Wide Web came in, it massified and democratized the sharing of written and visual information. The Internet of Things massified and democratized collection and sharing of data from all kinds of devices. And the new revolution is the Internet of Value, and that will massify and democratize exchange of value. So in simple terms, blockchain technology makes it possible for me to pay someone else money from my electronic wallet. It goes out of my wallet and into your wallet, and it's possible without a middleman. It's possible without a bank. In the past, banks were necessary to facilitate these transactions, and I think we are going into a new dispensation where that will not necessarily be the case. So the type of revolution that we saw when the Internet, the World Wide Web, and the Internet of Things were introduced, we are also now looking at the Internet of Value. So the combination of the World Wide Web, the Internet of Things, and the Internet of Value makes it possible for tourists to instantly offset their greenhouse gas footprint with carbon credits generated in communities around the Kruger Park in on an integrated digital ecosystem. This is my last slide. So the recent progress, Nova Institute has commenced with the feasibility testing of an end-to-end digital methodology for producing carbon credits. So the reason why we had to do that is that the current standards are moving too slowly in terms of the capability of actually transferring these types of credits on a blockchain, which is necessary to use the latest technology. So this methodology will enable the rapid issuance of locally produced digitally native carbon credits. So digitally native here means that the whole process takes place digitally. So the sensors that I referred to, and the monitoring and the evaluation, the transparency in clicking through to where the carbon credit is, those kinds of things is possible digitally now with the new technologies that's available. So what we will now do, and this is my last 4 bullets, is we hope to test and refine the end-to-end digital methodology. We've already started with that. We would like to issue these digitally native carbon credits. What is nice about these credits is the monitoring cycle is a lot quicker. And then we refine and test the tourist user interface. And lastly, or not lastly, it will have to, it's a constant process, but it will have to happen more seriously now. There should be some integration with the Kruger Park in terms of these specific type of things that we are testing. Thank you.
Thanks very much, Montague. Really interesting. I'm glad we got to see your slides in the end. And I think the take-home was that we're still allowed to braai. It's quite a relief. Thanks. So we're going to move on back to disease ecology. So Henriette is going to be speaking on behalf of Aisha and telling us about soil ecosystem diversity and anthrax. There was no time for questions for Montague, but if you want to grab him this evening or tomorrow, then please go ahead.
Move on to diseases again. So as indicated, I'm presenting on behalf of Aisha Asim, and I hope I do it justice. And it's in this presentation, we're addressing the soil ecosystem diversity, and we're trying to look at whether soil is the missing, or the intense investigation into soil is the missing puzzle in this disease. So, anthrax occurs in Kruger National Park, luckily for us, and it's caused by a bacteria, Bacillus anthracis. Now, in this diagram or this slide, we tried to indicate the factors involved in this disease. And if it looks complex, it is, so appreciate that. And also appreciate our enthusiasm to try and investigate all of these factors. So one thing about anthrax is it produces, it's a bacteria that produces endospores. And endospore is a form of, is bacteria that, not all bacteria produce it, and it enables the bacteria to survive in harsh conditions. So when an animal is infected and dies of anthrax, then normally scavengers open the carcass. So this is in Kruger. And then the exposure to oxygen then results in the bacteria, the vegetative cells, forming endospores. The vultures then feed on the carcass and then go and bath in water, so it's then spread. And one important thing about Bacillus anthracis, it is not contagious. So it can't spread from one animal to the next, and that's why all these vectors are involved. Then in Kruger, the main susceptible host was kudu always, and up until 1990. And then we saw impala becoming also a quite affected host. But with kudu, the blowfly plays an important role. So the infected carcass is opened, blowflies feed on it, they regurgitate the meal containing infected blood meal on leaves, and especially anthrax occurs in the northern part of Kruger, then where there's a lot of mpani trees, and then the kudu feeds on it and dies. Similarly, in this scenario where the animal dies, then the soil becomes infected again. So inoculum is created in the environment, and then through inhalation or ingestion, impala will then feed on grasses infected, and then the whole cycle repeats itself. But the most important thing about Bacillus anthracis is a soil-borne pathogen. So in many talks during this network session, a lot of people say we're just scratching the surface, and that is also the case here. So what happens below the surface? Because it's almost, we can observe stuff, but what is really happening below the surface? And in the soil, there is evidence that a rhizosphere interaction has been reported with Bacillus anthracis, amoeba, earthworms, and so forth, but there's not a lot known about it. And also there is evidence of genetic exchange within the soil between the spore formers, between the genus Bacillus. So I just want to bring you back to Kruger, and so we know that there's a vast diversity of Bacillus anthracis strains in Kruger. It's endemic in the northern part, so this indicates that it occurs there consistently. And then during large outbreaks, it will spread southwards towards the central part of Kruger. So the prevailing hypothesis is that the soils in endemic areas like the northern part in Pafuri it's creating a suitable environment. So there's high calcium content in these soils, and that supports the endospore to persist in that environment. And then that creates an alkaline pH, which allows the bacterial persistence. So to bring you back to how do we diagnose Bacillus anthracis, it's a very acute disease. So here I tried to picture, this is the bacteria, and its virulent factors are contained on two plasmids. And the one plasmid produces a PAG gene, we call it protective antigen gene, a protein actually, as well as on the same plasmid you get a lethal factor. Now, if the protective antigen and the lethal factor combines, it produces a lethal toxin, which then rapidly kills the animal. So in diagnosis, you not only look at microscopically at the bacteria under a microscope, obviously, but you have to detect these virulent factors using PCR. So there's been no record of Bacillus cereus biovaranthracis, but in primates in the rest of in Central Africa, they found Bacillus cereus with these two plasmids on it. So the question is, are we missing it if we're diagnosing it? Because we are just looking at under a microscope, we're looking at this typical, Bacillus anthracis make this typical box-shaped rods. And so are we missing other bacillus that is causing anthrax? So we use the Kruger surveillance system as a passive surveillance system where all rangers are given like a small pack where if they come across a carcass or any dead animal, they collect a smear on a microscope slide. And this slide is then sent to Skukuza State Veterinary Laboratory, and they investigate it using a microscopy. And you can see this system, or this passive surveillance system, is actually created for anthrax, so it's a little bit biased towards the north because everybody knows it occurs in the northern to central parts. But so this is the slides, or the smears that we now went back to investigate, and the red one was positive ones based on microscopy. So, So what we did is we took the smears and we used PCR to look at whether the lethal factor was there as well as the protective antigen. Because the assumption is if the lethal factor gene and the protective antigen is there, it must be Bacillus anthracis. So we looked at smears because there was a known outbreak in 2012, and you can see it actually persisted until 2015, and then it was just its normal occurrence in the endemic area. So the smears is the positive ones, and then we looked at Papa and Lev, which we presume is Bacillus anthracis. So the next slide shows it actually better. So here you can see, based on the smears, this is the amount of positive ones we identified, or the laboratory identified as anthrax. And those with Papa and Lev is very close to it. It's about 80%. So once again, it shows that you have to use both microscopy and all tools together, like with PCR, to make a diagnosis. But we have to investigate because there is some bacteria out there, or there's the presence of this Papa and Lev genes that was found that is not linked to Bacillus anthracis. So that needs to be investigated. And so we returned to soil, so we wanted to look at not only Bacillus anthracis in the soil, but also at spore formers. So Bacillus occurs in the Firmicutes phylum, this, I don't know what color it is, but that color there. And we collected soil samples from an endemic area, which is Pafuri, and Skukuza, which is a non-endemic area. And before we inoculated it with Bacillus anthracis, this is the Stern one, we just used the vaccine strain. You can see that those in Pafuri didn't contain any of these spore formers. It mainly contained the protobacteria. Skukuza was a little bit more diverse, although it had some. But the moment we inoculated with Bacillus anthracis, the whole microflora changed. So the question is, we're not looking at a microbial population, but what happens in an outbreak, and how does it change, and how does it influence one another? So, and especially, how does the spore how does Bacillus anthracis survival influence then the outbreak, and why does the outbreak occur? We also looked at soil properties. Like I said, there's this presumption that calcium-rich soils preserve the spores. And when we looked at the soil properties of soils collected in Pafuri and Skukuza, the calcium was actually not higher in Pafuri, but we've noticed that sodium, magnesium, and manganese, actually there's quite a difference between it. So it indicates that we'll have to go and look at these soil properties, because a soil-borne pathogen would like to be in the environment, and it prefers a specific soil. So we need to go and look at what soils does it prefer, and calcium is not a great indicator. What should we look at? Then there's this bacteriophages. So bacteriophages is a virus that infects bacteria. And there's been a link between bacteriophages and anthrax, or Bacillus anthracis. Well, first of all, bacteriophages are actually mechanisms of recombination for bacteria. So we know that they play a role in genetic exchange. But you also, with bacteriophages, you get phages that occurs more in a lytic rather than a lysogenic phase, or vice versa. And those that occurs in a lytic phase, what this phase does is the virus infects and replicates inside the bacteria and then allows the bacteria to burst. So it's a form of bioremediation, and we know that it occurs. So do they play a role in bioremediation? But what we observe is that the phages in the Pafuri soil was more abundant than those in Skukuza. So what we'll have to go and look at is we have to look deeper into bacteriophages. Are those that occur in Skukuza more lytic prone than those in Pafuri? So once again, how does this influence the spore survival and outbreak? Then the conclusion, are we closing the gaps? It feels like we're opening the gaps, but I think that is normal with any science. What I do realize is you have to look deeper, and I think the problem with such an environmental pathogen, and I think that is like, I'm a microbiologist, so I'm probably in the minority compared to ecologists here. But we are trying to investigate. As scientists, we are supposed to try and answer questions, and we're supposed to control variables. But in the environment, that is very difficult because there's so many variables. So I think what we have to go and look at a little bit deeper in soil composition and in chemistry. We'll have to also look deep into bacteriophages, and also how does the microflora influence one another, because in the end it's still like it's part of the environment, and also other factors that could contribute to it. And hopefully then we'll be able to close the gap, or maybe get a new question. This is all the funders, and thanks a lot for listening.
Thank you so much. We are running about 20 minutes late, so I'm going to ask you just to hold your questions for Henriette for afterwards. And our next speaker is Sunday Ochai, who's got a 5-minute slot. We'll be talking also about anthrax and transmission. So we're going to try and keep it to 20 minutes late. So we've still got about 4 presentations. So if we end and we're still only 20 minutes late, then I think that's a success.
Check. Hello, everyone. With this very, very extensive lecture, I believe by now we should all be converted. So it will suffice for me to say I will still be preaching to the converted. So I am Sunday Ochai, and on behalf of the co-authors, I'll be talking about the roles of host and environment in the transmission of anthrax. So anthrax, as has been said, is a zoonotic disease that affects a wide range of hosts, including humans, although the most susceptible species are herbivorous mammals. It has a global distribution, and Southern Africa, which includes the Kruger National Park, is considered the geographic origin of anthrax. And over the years, the northern part of Kruger is considered the area with the high incidence, while the southern part is considered to be anthrax-free. It is caused by a gram-positive endospore and capsule-forming bacterium Bacillus anthracis. And interestingly, over the years, as has been mentioned before, kudus have been the most susceptible host species or the most affected host species. But of recent, starting from, when I say recent, starting from 1992, it shifted to impala. And with this shift from kudu to impala, there was also an attendant shift from dry season to wet season event. Now we ask the question, could it be as a result of change in movement patterns? Could it be because of change in foraging behavior or modifications to their own foraging behavior? Or could it be the impact of climate or environmental factors? Could it be as a result of density? Or could it be the impact of other host species such as elephants? And to answer this question, we employ the services of this agent using the agent-based modeling and simulation, which is a computer simulation that we use to study the interactions between people, animals, things, places, and time. This is just to show you the logic that we use. So for the sake of this presentation, I will only be talking about the impact of movement on infectivity. This is to show you the logic and the parameterization of the model. Now here you can see, as I mentioned previously, that the northern part is the most, is where we have most of the cases. And here you can see with movement comes increase in number of cases. And then we try to just model 5 years, most especially for the sake of this presentation, comparing kudu and impala, and we realize that the first factor is that there is a relationship between movement patterns and infectivity. Also, we realize that where an animal dies determines which host species will be next affected. And lastly, we also realize that proximity to water holes was a significant determinant of which species dies and also the amount of animals that die. And also, with the increase in the number of years, we also saw an increase in the mortality rate. But one interesting thing for kudu is that in the last year, there was now a sharp drop. Now, anthrax is believed, or the spores of Bacillus anthracis are believed to last longer in the environment with decay starting after 10 years. While in this model, we saw that there was a decay just around the 5th year. Now, to answer further questions, we want to look at the impact of climate and environment, and also to look at trends regarding density, also to look at the impact of other hosts, like I mentioned, elephants. Could it be a direct impact or an indirect impact on the vegetation? And lastly, to see how the change in the environment also influences the roles of blowflies. With this, I want to thank all of you for listening. Thank you.
Thank you, Sanda. If there was a prize for time management, I think we would give it to you. Thanks. Please get hold of Sanda afterwards if you would like to chat to him about his work. So next up, we've got Carlo Andrea Cossol, who's going to speak about brucellosis in buffalo. Also a 5-minute slot.
Does it work? Good afternoon, everybody. I'm Carlo Andrea Cossu, and I'm a PhD student under the supervision of Professor Henriette van Heerden at the University of Pretoria under the Department of Veterinary Tropical Diseases. So here I'm going to present the partial results of our PhD, of our project, that is the isolation of Brucella abortus from African buffalo after 50 years in Kruger National Park, South Africa. Well, Brucella is a genus of gram-negative bacteria, small gram-negative bacteria, which at the moment we know that 12 recognized species that may occur in a wide variety of animal species, terrestrial mammals, marine mammals, and even amphibians. But Brucella actually causes a really important disease in livestock animals, which is called brucellosis, and actually cause abortions, stillbirths, and infertility in livestock animals, causing huge production and economic losses. Brucellosis has also an important zoonotic potential. It causes chronic and incapacitating zoonosis. And this is the reason why it is also classified as a biological weapon of category B. What is less known is also the effect of Brucella on translocation programs, especially for endangered species, which can hamper the outcome of these programs. For instance, early last century, the translocation of Blain's bison from Montana, USA, to the Hood Buffalo National Park in Canada, caused the importation of brucellosis, and after 100 years, the disease has not been eradicated fully, and it is a threat to the local Hood bison population. Well, we have quite some data, serological data from a wide variety of African wildlife, but actually serology is validated for livestock and not for African wildlife. So the interpretation from these results has several limits and limitations. Yeah. So actually, the gold standard nowadays, and since the last century, is still a bacteriological culture. And as you can see here, this is the only data we have from African wildlife based on bacteriological culture. And actually, the last isolation in these animals occurred in 1977. It actually, is when there was the first release of the Apple II computer, Star Wars. We had the champions from the Queen. It was also the first time that Argentina won the World Cup with Mario Kempes. Not Messi, not Maradona, Mario Kempes. And so you can understand that there is a serious gap of knowledge, of culture data, of bacteriological data. And when Brucella was isolated back in the days, they thought that it originated in livestock and then it spread to wildlife. But actually nowadays, different papers are highlighting that African wildlife is seen as a source for re-emergence of the disease, especially for livestock. So on these premises, we opportunistically collected tissue samples and serum samples from 105 wild African buffalo, Cape buffalo, from Kruger National Park, South Africa. And we tested for Brucella the buffaloes with a stepwise approach. We started with serological screening, and then confirmation with bacteriological culture, and finally with molecular characterization, with the aim to lay basis for understanding the epidemiology of brucellosis in Kruger National Park wildlife. So briefly, serological screening, we used two different techniques. So one was Ross Bengal test, which is based on agglutination. And then and the indirect ELISA, which is based on a colorimetric reaction, and then measured quantitatively with a spectrophotometer. Then confirmation was achieved by the culture of seropositive buffaloes from tissues of seropositive buffaloes on selective Farrell's and Sita media. And finally, we performed a gram staining to see if Brucella was present. And characterization was performed with Amos PCR and Bruce Ladder PCRs, which can identify up to 4 or 6 different species according to the technique. And you can see here the results of the serological screening. So we had 19 over 105 buffaloes that tested seropositive on RBT and ELISA. And here you can see the results of our culture. So you can see that we obtained by far 3 different cultures, 3 different isolates from lymph nodes and spleen. And molecular characterization was indicative of Brucella abortus in all the 3 cultures. So the main finding is the Brucella abortus isolation from buffalo after 50 years, nearly 50 years from the first and only isolation in South Africa. And open questions are, well, what is this? Is this a spillover from cattle? Or does Brucella abortus have a sylvatic cycle? Can it be maintained in the buffalo populations? Can it be spilled back to livestock? And moreover, what is the role or effect of brucellosis in wildlife? Can it affect other animal species? Are there other Brucella species more than abortus? And also, may predators be affected while feeding on buffalo carcasses? So next steps to clarify most of these questions, well, first is biotyping by biochemical methods, genotyping with different molecular techniques, and also attempting Brucella isolation from different wild animal species, not just buffalo, and also the investigation in livestock at the livestock-wildlife interface. So yeah, here are my contact details, and thanks for your kind attention.
Great, thank you so much. I'm sure everyone can approach you if they've got any questions. We're going to move straight on to our next speaker. Okay, so we're going to have Michael Clinchy talking about the fear of the human super predator. And is that a 5-minute slot? 15, no worries. You may have 15. Okay, and then we've got 4 talks after Michael, and then we've got our cheese and wine, just saying.
Right, good afternoon. The traditional view in predator-prey ecology has, until relatively recently, been that direct killing is the only way predators affect prey numbers. If there are 2 zebras and one is directly killed by lions, there is now 1 zebra. Now, the obviousness of this effect of direct killing is a big reason why the impacts of fear, that is, the anti-predator behavioral responses of prey, long remained unexplored in wildlife population and community ecology. Now, our lab focuses on experimentally testing the impacts of fear on wildlife population and community ecology, and to do this, we scare stuff. We conduct manipulative field experiments on free-living wildlife, which generally involve adding predators by simulating their presence using audio playbacks. We've experimentally demonstrated that fear itself can reduce wildlife populations by half in just 5 years, or just 4 years when transgenerational impacts are additionally considered. We've also experimentally demonstrated that fear of large carnivores can cause a trophic cascade by reducing mesocarnivore foraging, benefiting the mesocarnivore's prey, with cascading impacts on a competitor and prey of the mesocarnivore's prey. And we've also experimentally demonstrated that fear of humans can have landscape-scale impacts from mountain lions to mice, by causing avoidance and cautiousness in large carnivores and reduced activity in mesocarnivores, with cascading benefits for small mammals. Now, recent worldwide analysis have shown that humans kill carnivores and mega and mesoherbivores at much higher rates than they are killed by non-human predators. And from this, we can predict that fear of the human super predator is likely to exceed the fear of all other predators, given our greater lethality. Now, if fear of us is paramount across the planet, then we can further predict that the fear of humans is likely to cause the greatest ecological impacts. That is, impacts far greater than the population and community level impacts caused by the fear of non-human predators. So fear of humans could have wildlife populations in far less than 4 years and cause far greater cascading impacts than large carnivores in every ecosystem in the world. Okay, so how scary are we humans? Well, we've been conducting a series of comparable experiments across the globe to test this using these automated behavioral response systems we developed that integrate an off-the-shelf camera trap with a custom-built speaker. Now, all of these experiments involve testing reactions to playbacks of humans speaking calmly in locally used languages, dogs barking, and non-predator controls, and most also include large carnivore vocalizations. Our first experiment was on European badgers in the UK. We put buckets of peanuts mixed in sand outside badger burrows, and we demonstrated that fear of humans maximally affected latency to feed and multiple other measures, whereas fear of dogs and bears had lesser effects on latency alone, and hearing wolves had no effects. Next, we experimentally demonstrated that fear of humans caused cougars to flee from their own food caches and spend less total time feeding on those caches, whereas hearing dogs had no effects. After scaring cougars, we scared their prey, experimentally demonstrating that fear of humans caused white-tailed deer to flee more than twice as often as any other predator did, and caused the greatest reduction in overall feeding time at bait patches comprised of piles of corn. Moving to Sweden and working on fallow and roe deer, we experimentally demonstrated that fear of humans reduced the time deer spent feeding in agricultural crops significantly more so than hearing other predators did. Then, going down under to Tasmania, Australia, we experimentally demonstrated that fear of humans caused kangaroos and wallabies and other marsupials to flee from bait patches more than twice as often as did hearing Tasmanian devils, dogs, or wolves. And finally, we came here to Greater Kruger National Park, where we experimentally tested the reactions of the entire Savanna mammal community from elephants on down, which we were able to do because we finally got good at doing these experiments, partly by perfecting the equipment, amongst other things, and so succeeded in collecting an enormous amount of data, 4,238 independent trials involving 10,720 videos. So considering the community as a whole, wildlife here were twice as likely to run from humans than lions, whereas responses to hunting sounds, namely dogs barking and gunshots, were similar to lions. So let's think about this for a sec. We're more frightening than this, the world's most fearsome large carnivore, where it is most abundant, Kruger being home to one of the world's largest lion populations. Now, the experiment was conducted at waterholes during the dry season, and as a whole, wildlife here abandoned waterholes during the dry season in 40% faster time to humans than lions, whereas responses to dogs or gunshots were again similar to lions. Now, again, thinking about this in relation to lions, what this means is we're more frightening where the danger from them is greatest, both globally, i.e. in Kruger, and locally, at waterholes where lions do all their killing. Now, breaking these results down by species, greater fear of humans than lions was overwhelmingly pervasive throughout the savanna mammal community, as fully 95% of species either ran more from humans than lions or abandoned waterholes faster to humans. Now, not only was greater fear of humans overwhelmingly pervasive, it was also wholly unmitigated by other factors in most savanna mammal species based on a decision tree analysis. So whether it was day or night, whether the species was enormous or diminutive, or occurred in large groups or small, hearing humans was the sole best predictor of running in all but a small subset of species that rarely ever ran, for example, elephants. Now, considering each species separately,
my dad was a teacher, so I grew up in a home that the only thing I knew was sport.
Dit is baie moeilik om in Afrikaans te praat as daar net een persoon is wat Afrikaans praat en
Afrikaans is baie interessante taal. I'm baie respectful.
I don't know that I started loving the law. I think there were things that
Kudu?
I don't know that I started loving.
Because we have 11 official languages,
and Impala.
English is that one that everybody
all ran significantly more from humans than lions. Now, looking at the figure here, you notice that elephants rarely ran, as I've already mentioned, but they were also a significant exception in running more in response to lions than humans. And this is because elephants often ran toward the speaker or each other in response to lions, but never humans. Now, running toward each other, or bunching, is a defensive posture assumed by groups of elephants which may precede their attacking. So here we have an elephant that's going to hear a lion playback and run toward the speaker. A herd of elephants that has bunched upon hearing lions and then does this. And finally, an elephant that hears the lion and does this. One angry elephant. Now, remarkably, that camera actually did come back to life and survived. Now, turning to time to abandon the water hole, rhinoceroses and elephants
It's been a tough season. We played a lot of cricket, but we definitely always knew that the ultimate would be qualifying.
and hyenas, kudu, warthog, and impala all abandoned the water hole significantly faster to humans than lions. Now, focusing on elephants again, elephants abandoned the water hole faster to humans than lions, like most everything else, demonstrating that even elephants most fear the human superpredator. So there are several critical facets to the significance of this experiment, demonstrating that fear of the human superpredator far exceeds the fear of the king of beasts that bears reiterating. What it demonstrates is that fear of humans is more powerful and all-prevailing than the fear of the world's most fearsome large carnivore in one of the world's premier protected areas, possessing one of the world's largest lion populations at sites, i.e. water holes, where the danger from lions is greatest. Now, a further significant facet is that it demonstrates that the fear of specifically hearing human vocalizations far exceeded that of hearing hunting sounds, i.e. dogs barking or gunshots, just as elsewhere in all the other experiments around the globe I've shown you, indicating that wildlife everywhere recognize that humans are the predator, the real danger, while related disturbances like dogs barking are merely our lesser proxies. So in sum, we're accumulating growing experimental evidence that fear of the human superpredator is indeed paramount across the planet. Now, because the fear of us does appear to be globally paramount, then the impacts of the fear of us are likely greatest everywhere, far exceeding the fear of the population and community level impacts of the fear of non-human predators. And this would appear to add to the list of our worldwide environmental impacts, yay, humans, and may impede the photographic tourism conservation, particularly here in Africa, depends upon. Now, turning to applications, we and others have begun experimentally testing the feasibility of manipulating the fear of humans for conservation in the context of wildlife deterrence. In the experiment on deer in Sweden I mentioned previously, we demonstrated that playbacks of predator vocalizations significantly reduced crop damage and could thus be used to deter problem wildlife, thereby lessening lethal control. In a recent experiment in which Sam Ferrara and Adrian Schrader collaborated, they showed that playbacks of humans reduced visits by rhinos to middens in the same way we demonstrated that human playbacks deterred them from using water holes, pointing this potentially playbacks of humans potentially being used to help protect rhinos and other species by deterring their use of illegal hunting hotspots. On the flip side, another application we're exploring is facilitating wildlife habituation. So as I mentioned, if the fear of us impedes photographic tourism, that's bad news, particularly here in Africa, where gate receipts from photographic tourists are the primary funding source for conservation and the operation of parks like Kruger. Now, several African protected areas have successfully habituated gorillas and other primates to the presence of photographic tourists to help fund conservation, but it can take years to accomplish this habituation. Now, in the experiments on populations and communities I described at the outset, we went to great lengths to avoid habituation by broadcasting infrequently and irregularly. If we instead broadcast intensively and predictably, then potentially playbacks of humans broadcast at carefully regulated locations could be used to aid conservation by helping to rapidly and cheaply habituate wildlife to the presence of photographic tourists. Now, we couldn't do all these experiments around the globe without tons of excellent collaborators, some of whom are in the room, and our thanks to them all, and our thanks to you very much for listening.
Thanks. Yeah, really, really interesting. So you were actually 2 minutes under. So I think we can allow one question. Is there anybody? Oh, there we go. You had your hand up first, so I'll bring you the mic.
So I saw at the end there that there was one paper on song sparrows. Do you have, or have you looked at the effects of human playback on other taxa besides mostly mammals?
Not yet. How's that for a quick question? Quick answer.
Are you satisfied?
Do you have any plans to do that?
We have plans to do a lot of things, but everything takes time. Cool. Quick answer.
Okay, I think we'll leave it at that. Thanks very much. So we're going to go on to our next speaker. Okay, so our next speaker is Michael Anderson. He's going to be talking about body size dependent succession patterns in the Serengeti. And we're going to minus 2 minutes off your talk because you handed your presentation in late. I'm just joking, we're not.
Can you guys hear? Yeah, can you hear me? All right, now I know why. If everybody goes bolting out of the room, now I know why, thanks to Mike. All these scary humans are looking out at me. So I'm going to talk today about competition and facilitation driving migration patterns in Serengeti ecosystem, but I'd like to start by thanking this incredible team of collaborators. Really a huge effort went into what I'm going to talk about today, and we couldn't have done it without just terrific collaborations from everyone on that list, so I'd like to thank them. So this picture is showing wildebeest migrating through the Serengeti Plains. It's an incredible event that happens annually, and arguably it's the defining feature of the Serengeti ecosystem. It's also likely, historically, the defining feature of many grazing ecosystems around the world that no longer have these migrations, Kruger National Park likely being one of them. But in Serengeti, this migration is composed of roughly 200,000 zebras, somewhere over 1 million wildebeests, and over 400,000 Thompson's gazelles. So what do we know about this migration? Well, if we look back historically in the literature, Gwen and Bell in the late 60s suggested that there was significant variation in consumption by these herbivores, in which zebra tended to eat the tops of grasses as they migrated through the veld. Wildebeest tended to focus on the leaves, and Thompson's gazelles on the sheaths. I simply note that this came from a relatively small number of samples, just 6 individuals in the wet season and 14 in the dry season. Okay, what else do we know? Bell documented from a single plot that he monitored for 3 years in the western corridor of Serengeti. He suggested that herbivores, small herbivores, follow larger herbivores in time, such that you get zebra coming first, followed by wildebeest, followed by Thompson's gazelles, and that was published in this one of my favorite papers, this 1971 paper in Scientific American. What else do we know? We know that smaller herbivores tend to follow larger herbivores in space. This is a paper that was published by Sam McNaughton in 1976 in Science. And what he found is that after a huge accumulation and buildup of wildebeest in the Moru Kopis area of Serengeti, this was followed by the subsequent grazing by Thompson's gazelles roughly 3 to 4 weeks after. And these observations led to the idea that this ecosystem is really, we see a grazing succession in this ecosystem, and that the migration follows this grazing ecosystem. And what that means is that herbivores of varying body size use a site over time, and then they modify the vegetation at that site that actually changes the vegetation for the herbivores that come after. And like plant facilitation, The idea is that early successional species do some modification of the environment that facilitates the later species, the later colonists. And so the way this would work in grazing succession is that larger-bodied herbivores modify the vegetation and make it increase the forage quality for herbivores, smaller herbivores that come after. But we argue that significant questions remain. Pennycook in 1975 did some analyses of aerial census data suggesting that wildebeest and zebra may not actually separate themselves in space and time. This was conducted at the grain of 5 kilometers squared. In 1985, Tony Sinclair published an excellent paper actually challenging the idea of facilitation, suggesting that it was competition that underlied the spatial and temporal patterns of this migration. And what he argued is that because these animals are constrained by their jaw and muzzle size to forage at different heights, and smaller herbivores could graze the grass down lower than the larger herbivores, it may be that the smaller herbivores are actually outcompeting the larger herbivores and pushing them ahead and forcing them to move forward. This was further suggested by Ilias and Gordon and Dutoit and Olf in 2014, and there's substantial evidence for this. I just chose this paper, Arsenault, Nolan Smith in 2008, looked at where these animals use and get their forage, and there's wildebeest tend to forage at a much lower level than do zebra, which tend to use much higher, forage much higher in the grass field. Okay, so the overall goal of our study was to investigate grazing succession of migratory herbivores in Serengeti, and we wanted to ask, does it occur at all? And if so, what are the underlying drivers of herbivore movement patterns? So our first aim was to use spatial occupancy modeling in order to look at interactions between animals. And so what we did is we estimated conditional probabilities from a multi-species Bayesian occupancy model. That's kind of a mouthful. But that controlled for spatial and temporal autocorrelation. And we expected, if facilitation is happening, we expect largely positive effects from larger to smaller herbivores, and if competition is occurring, we expect largely negative effects from smaller herbivores on larger herbivores. Our second aim was to use interannual variation in rainfall and fire in order to test hypotheses about species interactions. And the idea here is that changes in the grass quality and quantity should alter the strength of herbivore species associations during migration. So if facilitation is occurring, rainfall should strengthen positive species associations and fire should weaken them. And likewise, competition is operating, rainfall should weaken negative species associations and fire should strengthen them. Our third aim was to analyze movement patterns in high and low rainfall years to better resolve species interactions. And the way we did that is we had known animal densities with animals that we could track in space and time, for example, using GPS collars. And then we tracked how they moved in relation to dense heterospecific herds. If facilitation was operating, we expected larger migrants to move independently of small migrants and smaller migrants to track larger migrants through time, but more so in wet years than dry years. And likewise, if competition was operating, we expected smaller migrants to move independently of larger migrants, and larger migrants to alter their distance and step length away from smaller migrants, especially during dry, low grass biomass years. So our fourth and final aim was to look at, to analyze diet and quantify diet patterns to better resolve the degree of competition versus facilitation. The idea being that if there's a lot of dietary overlap, there has to be a lot of dietary overlap if competition is occurring, whereas if dietary overlap is low, either competition is not occurring or there's significant resource partitioning. All right, so how did we do this? Well, we analyzed 8 years of camera trap data to study large-scale herbivore migration patterns. This accounted for environmental heterogeneity, autocorrelation in space and time, and it was done at a landscape scale. We used data from 2011 to 2018 from the Snapshot Serengeti project, which is over 200 cameras in the central area of Serengeti. We also used 2 years of GPS collar data used to study the migration patterns of wildebeest and zebra as they moved through the Snapshot Serengeti camera area, shown here. And these controlled for fine-scale movements of individual herbivores and allowed us to look at species-specific responses. And then we collected 2 years of fecal DNA barcoding data. These were used to study dietary patterns, and we sought an approach that accounted for diet variation in time and space and allowed us to separate diet by species. Okay, so what were the results we saw? Well, the first result we saw was that Thompson's gazelles tended to lag behind zebra and wildebeest by roughly 1 month. And no, Nikki Stevens, I don't expect you to read all these little tiny, this text here. I'm going to blow this up for you because I know your eyes are going. So here's, I'm going to show you a few different years of what this looked like. This is the total number of counts of animals in the grid as they migrate in different years during the transition season. And what we see, like in 2011, here the black line is zebra, the gray line is wildebeest, and the brown line is Thompson's gazelles. You see co-migration of the 2 larger herbivores and then spacing by about a month. I'm simply going to show you up here. If you look at this grid, this shows you the amount of fire. More red means more fire. So this is an example from 2013, a very high fire year. The animals disperse a lot more, and there's much less of a strong positive association. And then here's an example of 2018, another relatively low fire year, and a lot of co-migration and overlap between wildebeest and zebra, and then about a 1-month spacing of Thompson's gazelles. So we looked at these interaction strengths from the Bayesian occupancy models, and I'm not going to really go into the details, but what we did is we looked at the residuals from the mean and said, was it a strong year where there was positive association between zebra and wildebeest or negative? And we took all these residuals and plugged them into regressions and looked at the effects of rainfall and fire. And what we found was something pretty striking, is that as burned area went up, the effect of large species on smaller species really went down, and with rainfall it went up, but only from larger species to Thompson's gazelle. There was never and I'll show you the next result, which is for, this is for zebra, this is for wildebeest. We essentially see the same thing. There's a strong negative effect here for fire, and a really strong positive effect here for rainfall, and never really an interaction here with respect to rainfall for zebra and wildebeest. And I'm not going to show Thompson's gazelles because there were no significant effects of Thompson's gazelles on either zebra or wildebeest. Now, looking at the GPS collar data was really interesting. Wildebeest did not respond to heterospecifics in any way, whereas when we looked at zebra, we found a really interesting result, that the distance from large herds of wildebeest significantly predicted their spatial movement patterns. And what was interesting is in a dry year, they spaced themselves much further away from large herds than in a wet year. So they're spacing themselves in both years, but they had less of a buffer in a wet year when there's lots of resources around. The final thing we saw was really strong resource partitioning in the diet. So essentially the more colors in these pie charts is just more different things. Green is represented by grass. This is for 2017 and 2018. Thompson's gazelles had an incredibly diverse diet. And what was really interesting is when we analyzed this through time, we found that the proportion of Poaceae, or the amount of grass eaten by Thompson's gazelles, was low throughout much of the season and then really increased at the end of the dry season. These data are for wildebeest, and these data are for zebra. We dove into this a little bit more and found that what the Thompson's gazelles were doing were switching to the very high quality grasses like Digitaria macroblefera and those in the PACMAD clade. And what was even more interesting about this is these were the species that had been selected and been grazed by wildebeest and zebra 1 month earlier. So this was really interesting evidence that they were foraging on forbs and perhaps tracking wildebeest and zebra because they were opening up the grass layer and giving them access to forbs, and then later in the dry season they were actually switching to the grasses that had been previously fed on by wildebeest and zebra. All right, so what are the conclusions? The data support classic grazing succession for Thompson's gazelles, but not at all for wildebeest and zebra, which tend to co-migrate. Rainfall and fire regulate the strength of interactions between these migrants across years. Occupancy and GPS collar data reveal competitive processes with zebra being pushed ahead of wildebeest. And then finally, diet data suggests competition between large herbivores and facilitation between zebra and wildebeest, and then the smaller Thompson's gazelles. And so just to conclude, this led us to put out or conclude a novel hypothesis about the way the Serengeti migration is operating. We call it the push-pull mechanism, in which zebra are pushed ahead by wildebeest by competition for grass, and then Thompson's gazelles are pulled from behind by foraging facilitation, forbs early in the season, and then grasses later in the season. And again, this was a tremendous effort for many people, and I would just like to thank everybody who participated. Thanks for your time.
Great, thanks, Michael. Super interesting. Really nice slides. Thanks very much for an excellent talk. There is a minute. Are there any hands? I'm going to give you a few seconds. There's one hand. Let's take one question, if you don't mind. You can use this mic. Is there a mic there? It's coming. The mic is coming.
Can you hear me? Thanks, Michael. Just a quick question about it. You know, Bell's idea is fantastic until you look at the fact that you have regrowth, and so I wonder if a month after zebra and wildebeest are coming through, it's not opening up, but it's a flush of regrowth off, because once grass gets hammered, it comes back. And what they do is they move into an area with lots of nice green, low-fiber grass. I mean, is that a possibility?
Yeah, so if my slides were still up there, I would take you just a few slides past in our camera trap grid, which shows after the wildebeest go through, even up to 1 month after, there's incredible access and visibility of this forb layer. And I think early on in the migration, that's what they're tracking. I mean, I think that's what our data suggest. And I think the regrowth is really important, and that flush, I think, if I'm understanding your question, I think that's what we picked up when we see the switch to grasses in that late part of the dry season. I think it's the regrowing Digitaria and those in the Pac-Mad clade that were previously grazed by those animals that they're foraging on, and when the forbs are all gone, because that's a late season, a dry season phenomenon. Yeah, does that answer your question?
Yeah, and that's exactly what I was thinking.
Yeah.
Cool.
Thanks.
Thanks so much. Okay, so we're going to move straight on to Richard. Richard Gibbs is going to be sticking with drivers of animal movement.
All right, thank you, Louise. Hi, all. There we go. I am a second-year PhD candidate at Stellenbosch University, part of the Mathematical Biosciences Hub. And today I'm basically going through, give an overview of my PhD, really, and how we think adaptive and subjective perceptions drive individual animal movement behaviors. I should preface this with the fact that we're considering large terrestrial mammals with a relatively high level of cognition, and at the individual level. So I'm sure for this group I don't need to go too much into the importance of movement, but in a sentence or two, the movement behavior of an animal closely interacts with individual fitness and affects a myriad of ecological processes. And it can also be considered a key conservation metric for certain species, whether this be species at risk or just influential to the rest of the environment. Now, an animal's movement behavior must efficiently fulfill multiple goals at the same time, and it does do this. It's remarkably good at doing this. And previously, this has led people to believe that they behave optimally, and we've predicted behavior by looking at optimal foraging techniques or that kind of thing. But this efficiency doesn't mean that the animal has a complete or even accurate understanding of its landscape. Rather, it's based on a number of heuristics. And rather, behavior is the result of the current goals of the animal as well as its subjective perception of the environment. And as I say, this perception could be subject to numerous flaws or biases. And these are the things that we'd like to highlight in our considerations. So, of course, there are a number of ways that we can study an animal's behavior. At the base level, you can look at the fundamental abilities of the animal, so whether that be the sensory techniques and capabilities of the animal, or on the neurological level, you can look at their navigational capacities, their spatial awareness, or memory capabilities. On the other side, you've got the ecologists can look at the movement patterns, the realized behaviors of the realized patterns of movement. And that can be different movement styles, such as the use of repeated parts or shortcuts. They can look at how adaptable the patterns are to various stimuli, or you can look at the terminal home range design and the attracting habitats, favored resources, and whatnot. I know I'm grossly downplaying the ecological side of it, but just bear with me, because the point I'm trying to make is that between these two observable methods of looking at movement behavior, there's this the hidden behavioral mechanisms that drive an animal's movement behavior. And this is very hard to nail down. We can't really encapsulate it. But if we could encapsulate it and reproduce it to a certain level, this would increase our understanding of a movement behavior greatly. And if we can build our understanding of an animal's movement behavior in this bottom-up manner, it will allow us to extend our understanding of movement behavior to novel landscapes, novel situations. So whether that be transplantation of an animal into an entirely new landscape, or it could be the just alterations to the landscape, so changes in fencing, artificial watering holes, or conservation corridors. So we could more meaningfully predict how the animal would behave in those situations. Like I say, nailing down these rules is difficult. It's hard to test and it's rather messy, but that's what we're working towards. So I'm a mathematician by training, primarily, and the benefits of using deterministic mathematical models to look at these kind of problems is that they allow us to examine the problems in a mechanistic and more rule-based manner, which is exactly what we think that these behavioral mechanisms could be. Unlike your more traditional statistical techniques, they're very tractable. You can play around with them and use this to kind of determine the fundamental laws and the interactions between the various components of what you're considering. So whether that be how an animal's abilities are affected by its environment or whatnot. And as I said, I'd just like to reiterate as much as I can that it would allow us to extend our predictions of movement behavior into novel situations. The main drawback is that they do require more sophisticated means of analysis, and this is especially true when you're considering real-world messy data, because they're very sensitive, and you have to take that into account. So I'd just quickly like to highlight two features before I get on to the more interesting, the model, which is more exciting for me. And that is that there is evidence both from the neurosciences and from the ecological observations that animals are able to maintain some kind of a map-like representation of their environment in their memory. This is obviously limited by their memory capabilities and certain environmental features such as changes in the environment. And this map must be constantly adapted and updated in order for the animal to maintain a relevant understanding of its environment. The drivers of movement are also variable, and this could be dependent on the state of the animal or on seasonality or the life stage of the animal. So now onto the model itself. What we've got is an individual with a certain sensory ability, and as it moves through the landscape, it's able to sense, we're still looking at how it's sensing the environment. We'd like to put a bit more consideration into that, but it's able to build its representation from its sensory abilities as it moves through the landscape. This has been done before, the cognitive map modeling techniques, but what we've done is also add this layer of certainty for the various drivers of movement. And this, we're hoping this will allow us to look at more of the exploratory nature of novel situations. Both of these maps will decay over time and smear as well to account for memory loss and locational degradation and whatnot. And both maps are updated as locations are visited. So the memory map will be updated with newer idea of the values there. And if this differs from what the animal recalls, then the level of certainty of that for that variable in that location will decrease. So together, these two maps, So together, these two maps will give the perceived distribution of one resource of a notable variable that's affecting movement. Well, this is more of the long-term perception because this combined with the sensory ability will give the full perception of that variable. And notable variables can include resources, the landscape of fear, or very importantly, the physical energy landscape, so how well the landscape can facilitate the animal's movement, so the ground cover and gradient and whatnot. So now we've got this adaptive map of perception, and previously studies have taken their maps and just condensed them into one thing by looking at the average of the relative amount that they affect movement. But what we'd like to do is take into account the fact that the drivers of movement change through time as well, following notable ecological processes. So here, I'm sure it's pretty small, but I'm just looking at hunger and time since it was last fed, so this is more, so these change over various timescales. This can also be seasonal and whatnot, and we'd like to be able to include how these drivers change in our analysis of the movement behavior. And so all of the drivers and all of the maps at a given time can be condensed into one combined landscape of movement, movement facilitation. And in a nutshell, that's basically the model that we're looking at. So I'm just going to quickly finish off by running through the procedure that we're going to look at, how we're going to look at this model. So first step is obviously analyzing the model. And firstly, we're just going to see if we can replicate some observed behaviors, some notable observable behaviors. But then this analysis must also include looking at the various functions that we're considering and the behavior of the different parameters, how they interact with each other, and the interaction between the animal and the environment and whatnot. And this is where we kind of can determine the governing laws and relationships between the various components, and that's where we can get the more theoretical understanding of what's going on, which could still be useful. But the second step of this is to determine a data fitting protocol, and that's where we try and find, basically just trying to find the best way to fit data to our model. So we'll take some of the data that the model has generated and then try and reverse engineer it to get back to the parameter values and an adequate representation of the behavior. So this would be more the traditional statistical sampling methods and your Monte Carlo methods and whatnot, as well as possibly machine learning. We'll be just going to see what works best. And then if this goes well, this is where it will get interesting. If this goes well, we can finally get to the point where we fit real-world data. And this is, yes, I have to premise it by saying that's if these two steps go well, but this is where I'm really pushing towards, and this is what I'd like to get towards. And that is where we consider more, in our model, add more of the species-specific attributes and experiences that they are subject to, and then take your telemetry data for the movement as well as remote sensing data for the various environmental variables, and try and fit this to our model. So this is obviously more of a direct interest to landscape managers because it could give you these quantitative predictions. It could be a very good predictive tool for how this could change in novel situations. So, I mean, if you're considering the design of a corridor or something, there's obviously other factors that affect this. So if you'd like to try and find the best possible way forward, we can try and predict how the animal will behave at the placement of corridors in different places, and then you can leverage that against the different locations that you're looking at. As also, the parameters, if we were able to successfully fit these parameters to the model, they could be quite important drivers, quite important to indicate the drivers of movement in your specific landscape. So this is very landscape specific, so that could be quite useful as well. This, of course, would depend on the resolution of the data and the grain of it. But even if we can only kind of fit for the terminal space use of the animal, I feel like it could be quite a decent tool and quite useful to conservation efforts. And yeah, I'm hoping I've left enough time for questions. And yeah, please ask ahead if you have any.
Okay, thanks so much, Richard. So I was told that I'm not allowed to have any more questions, but has anyone got a question? One, quick one? Okay, go on. Thank you. Please grab Richard at the Cheese and Wine if you've got any comments. I think there's some really nice potential with some of those models. So the next speaker is Norman Owen-Smith, who is going to be talking to us about geology and the relationship with geology and herbivores. And then we've got one more 5-minute presentation after that. So let's just give the speakers a chance to share their research with us, and then we'll be finishing up soon.
Thank you. Okay, this is going to be a somewhat different talk to those that have gone before, because it was intended as an introduction to the workshop that we held in conjunction with this meeting, called the African Soils Workshop. We had expected that the workshop would take place after the Savanna Science meeting, but for various reasons it took place beforehand. So I'm now introducing that workshop, but seeing most of you weren't able to attend it, it'll indicate why we organized this workshop. It's intended around the topic of soil fertility and what actually governs soil fertility. It's the aspect of ecology that I recognize as being important, but which I understand least. What is it that defines soil fertility, not for growing crops, but for feeding herbivores? And it's based on an idea put forward by a guy whose name you've seen up here before, Richard Bell, that there's a relationship between bedrock geology, soil fertility, plant palatability for herbivores, and the abundance of those herbivores attained in the area. Richard Bell was an interesting character. We already heard about his pioneering work on grazing successions up and down the landscape. After he'd finished with his Serengeti study, he was then involved in surveying wildlife populations around Africa. And he was struck by the huge contrast between the hordes of animals you saw on the screen in the Serengeti and the fact that the large parts of Africa that you fly over and see very few herbivores in it. As we in the South had always maintained, all of Africa is not like the Serengeti. And the idea he put forward there, Richard Bell, he put forward that there's the contrast between different parts of Africa, as I mentioned, and he used the status of the bedrock as a surrogate for its likely nutritional status for herbivores. He assumed that volcanic substrates and Rift Valley sediments would yield more fertile soils than basement granite or gneiss and other kinds of sediments. And that would underlie the huge difference in herbivore abundance. I met Richard Bell when he gave that talk at Oxford, and it was at his invitation that he then came out to speak at the workshop that formed the basis of the book called The Ecology of Tropical Savannas. The article from which I extracted the quotes was from his chapter in that book. Brian Huntley was one of the organizers of the meeting, a very astute ecologist, but he approached things from the plant side. He was a plant ecologist. He'd just come back from working in Angola and was likewise struck by the large areas in Angola that are underlaid by Kalahari sands of very low nutrient status, and the very big difference between the abundance of herbivores on those regions of Angola and what you might see in more areas of more fertile soils. And he then drew a distinction between mesic dystrophic savannas, mesic in terms of rainfall, and poor in terms of the nutritional value of soils, and arid eutrophic savannas. So in addition to the effects of rainfall on both food for herbivores and the quality of vegetation, there was also an effect of soil fertility, and he suggested it was related to the base saturation of the soils, but didn't make a link to geology. That's a huge welwitschia that he's standing next to in Angola in that picture. And his book on the biodiversity of Angola has just come out. I'm sure it's going to be fascinating to read, but you have to have quite a deep pocket to purchase it. But it may be available free online under some conditions. Oh, I forgot to mention the sweet-filled flower-filled distinction. The botanical distinction that Brian made was between fine-leaved and broad-leaved savannas. The fine-leaved savannas are the acacias, name we still use. They're defended by thorns. And the broad-leaved savannas actually occur in two somewhat distinct forms: the Miombo woodlands, dominated by Brachystegia, and the Sudanian woodlands, dominated by Combretum species, Combretaceae, Combretum terminarius. And then Russell East expanded that analysis to look at the responses of individual herbivore species to soil nutrient status within the context of Richard Bell's classification. In other words, he just simply used Richard Bell's classification of regions in terms of the underlying geology and didn't use any measurements for soil fertility because they weren't available to him. Bob Scholes then synthesized these views on soil influences upon African savannas and emphasized that they influenced almost everything in those savannas: the herbaceous production, the vegetation composition, plant morphology, forage quality, herbivory, and that the biological activities also fed back into the vegetation to also affect its effective nutritional value. So everything seemed neatly synthesized. The final clinching paper was written by Hervé Fritz and Patrick Duncan, who actually then got the data, more detailed data on herbivore abundances and put that into a paper along with bedrock geology and produced the curve you see on the right of an increase in herbivore abundance with increased rainfall, but two parallel lines. The one line related to areas underlaid by bedrock that produced fertile soils, and the other underlaid by bedrock that produced much less fertile soils and supported fewer ungulates. I then came to synthesize what we know about African ecology in my book, and in putting it together, the data, and looking for the basis for that generalization, I started to find exceptions. And as is often in ecology, not that many rules apply globally. Nothing works all the time in ecology, because ecology is all about how plants and animals behave, depends upon the environment, and different things might come out in different environments. So generalizations might have to be contingent. There's already a contingency in the Fritz and Dunker's regression there, because they assumed that that relationship only applied over a certain rainfall range between 500 millimeters and 1,000 millimeters. Above 1,000 millimeters, soils were leached, whatever the substrate, and therefore the soils were generally nutrient poor. Below 500 millimeters, there was almost no leaching, and the soils were then mostly nutrient rich. So first contingency only valid for intermediate rainfall ranges, but of course that is the rainfall range applicable to most of savanna Africa. The exceptions are in parts of West Africa that have higher rainfall and wetter savannas like South America and tropical Asia. There's my plot trying to establish the link between the bedrock geology on the left and a measure of soil fertility in terms of cation exchange capacity on the right. And the first thing you can see on the left is that the highest large herbivore biomass recorded anywhere in Africa came from Uganda, dominated Murchison Falls, coupled with Queen Elizabeth Park, dominated by elephants and hippos, these two mega herbivores. And the soils there are not fertile soils, they are Rift Valley sediments, but there's no volcanic influence. Next in line in terms of herbivore biomass is HRP, just pipping Serengeti. And then you get down at the far right end to the Miamba woodlands, represented not only in West Africa with these soils, but also in Malawi. And of course, the Miamba woodlands also extend to parts of Eastern Africa, through Zimbabwe, Zambia, and into Tanzania. So you have to, and elephants are abundant through most of that range until you get to Miamba woodlands, but elsewhere you have to take elephants off their pedestal. Elephants don't seem generally to respond to soil fertility. And hippos also not, but that's probably because hippos are feeding mainly near rivers, and that's where sedimentary soils form. So, we now set out in our workshop to now pull the data to make the links. These are some of the contingencies to take into mind. It only applies in a certain rainfall range. Are those geological distinctions the ones that are valid for affecting soil fertility? How do you measure soil fertility? Is cation exchange capacity the appropriate measure? Because those cations are not what the herbivores seek. Phosphorus is probably the major limiting nutrient for herbivores, and nitrogen is probably the main influence for soil fertility, but nitrogen does not come out of the bedrock. It's contributed by plant organic matter that decomposes and recycles nitrogen back into the soil in the form of various proteins. Vegetation types, there's a distinction between fine-leaved woody plants and broad-leaved woody plants. But what are the distinctions among the grasses? Because you're looking primarily at the abundance of grazers that make most of the large herbivore biomass. Those grassland distinctions are not quite so clear, they're a bit opaque. And then the other issue is which herbivores? Do all herbivores globally respond in the same way to soil fertility, or is it only certain grazers? So these are our questions to be addressed during the workshop. Firstly, how does bedrock geology influence soil fertility for large herbivores? In other words, which components of the bedrock, there are various forms of volcanic substrates, there are also various forms of granite. Which forms of bedrock substrate influence which measures of soil fertility for large herbivores? Is it simply soil texture as it feeds into cation exchange capacity or base saturation, or is it something independently affecting the phosphorus availability of soils for those herbivores, which also influences plant growth, but not as strongly as nitrogen and potassium? How does soil fertility influence the nutritional value of savanna trees and grasses? How would you measure nutritional value? Most people measure protein content, but generally plants are quite adequate in protein content, and it's the digestibility that's more important. But mineral nutrients may also come into play, but again, it's not only phosphorus, but also sodium, crucially important for herbivores. And sodium is more localized and isn't generally a component of the bedrock. So certain rocks may be have more sodium available, and others less. And then looking at things botanically, what are the soil features that distinguish eutrophic from dystrophic savannas? Is it simply clay content? Because many of the cases are associated with clay, but not all of them. Not that simple. Which large herbivores respond to the soil fertility distinctions? Elephants don't. What else doesn't? Grazers also respond similarly to grazers, and all grazers respond the same way. Probably not. And then how do large herbivores modify the effects of soil availability? Not only by creating grazing lawns, which only certain herbivores do, but also by grazing down grass and therefore preventing that grass being burnt in grass fires. That has fundamental influences on nutrient cycling, where the grass is grazed in the dry season or left standing and eventually burnt by fires. And then finally, important thing for ecosystem functioning, how does all of this affect the proportion of the vegetation that is recycled rapidly through fermentation in a ruminant's gut or non-ruminant's hind gut, and what portion of the vegetation does not get burned and falls to the soil and yields its nutrient contents much more slowly through decomposition by termites and other soil microorganisms. Those are the questions. Next year we hope to have some answers for you. Thank you for listening.
Thanks, Norman. Some really interesting questions. We look forward to next year. So we're going to move on to our last speaker for today. Before we do that, just want to say thanks to the sound guys. There's been a few technical challenges, and I think I'd rather be up there giving a talk than being in charge of presentations and sound. So thanks for holding it together for us. And we're going to go straight into Michael's talk. Michael will be talking about tracking hippo. Not so easy, apparently.
Can you hear me? Great. Great. Hello, everyone. Afternoon. Well done for making it this far and to the last session and talk of the day. I'm just going to share with you some progress that I've made on one of my PhD projects. And this is sort of a tagging and tracking approach, and with the big idea of contributing towards knowledge on the ecology of one of Africa's mega grazers. So I think many of us are aware of the diverse influence that hippos have in the ecosystem, ranging from their bulk grazing to their nutrient transfer between land and water, as well as their trampling and the mechanical action of their wallowing in both terrestrial and aquatic ecosystems. But other than being aware of these diverse array of impacts, we really have a limited understanding of the spatial and temporal nature of hippo ecosystem engineering. And this is largely because hippos are cryptic. Their influence on the environment is cryptic. They're nocturnal, for one, and obviously semi-aquatic, which makes their effects largely difficult to visualize and observe. So for this reason, an approach that could investigate and look at hippo movements could really facilitate a better understanding of their role in the environment, as well as being applicable to a variety of applied conservation-related settings. And so it's on this background that we decided to investigate a tagging and tracking approach for hippo. But obviously, there's a reason why we know so little about hippos and why there's very limited knowledge on hippo movements, and that's because hippos are highly sensitive to the opioid-containing drugs that are traditionally used for their immobilization. And another sort of facet of that is that there's a potential for drowning once they've been immobilized. Once immobilized and heading straight to water, they can drown. And there's the obvious one that it's really a dangerous species to work alongside, whether you're a veterinarian or a handler. And so Keenan Steers is one of the few chaps that's been able to do work using ankle collars, and that's the only real published study that we have on hippo movements. And altogether, these sort of ankle collar approaches are also very resource intensive, other than being sort of a risky, dangerous approach. So this was our rationale for trying a remote tracking device deployment technique. And this is a technique that uses a crossbow that fires a small little limpet-style device that has two medical-grade titanium barbs on the end. This has been widely used in the study of cetaceans, whales, and dolphins, and it has led to some considerable insight, for example, on the movement of killer whales in the Southern Ocean. And so we set out to try this on hippos. So our first port of call was to try this approach on hippo carcasses, just to do our due diligence. And we wanted to test, number one, which region of the body would be best for this, either the hind rump or the neck, and this could have been useful for accelerometer use. We tested two barb lengths that are commercially available, 47 and 69 millimeters long. And then we also tested the distance away from the animal that we need to be in order to get adequate penetration of these barbs. And so it's just a snapshot of what we found. We found that the hind rump would be the best location for these devices. And this is primarily because of the additional fat layer that hippos have beyond their skin. So that could provide protection of the barbs against the muscle tissue. We found that short barbs of around 47 millimeter barb length was the most appropriate because it didn't reach into the muscle, and that 15 meters away was sort of the maximum distance we could be in order to get adequate penetration of the barbs. And we worked on a site on the Crocodile River to try this now on live animals. So we took what we learned on this carcass trial, and we tried it on live animals in the Crocodile River. And I'm going to show you a quick video of what this entails. And you can see I'm on a safe, an elevated location here, and using this crossbow, I deploy it into its hind rump. So it's not for the faint-hearted, so I wouldn't do this at home. But certainly we've had a lot of experience with doing this on the killer whale side of things, so it's a relatively safe option with the right backup and support crew. So the next step was the data retrieval, and this has been our biggest challenge so far. We've moved from using a handheld UHF receiver to a LoRa tower now, which is assisted with us, but we're still in the sort of development phase of these tags. But I was going to share with you just a quick little movement track that doesn't seem much, but I'm pretty chuffed with this. Just a glimpse into what these hippos are doing. We know so little. So this is a hippo moving from the Crocodile River north, venturing to this pan that I've shown on the right here, about 4 or 5 kilometers away, and then returning the next morning to the river. So I found that quite satisfying. And then I think sort of next steps in this project is continued device development. These tags really take a beating, as you can imagine. So we're going to keep working on that with a crowd that's been assisting us with that. Data retrieval is obviously a big challenge. So we're moving away from sort of basic UHF to LoRa systems and potentially satellite later on. And then sort of to refine this approach really requires continuous learning. And sort of the major goal of all of this is to inform space use of hippos, to inform us on their ecology, and I'm hoping this can be an approach that could also be used to investigate interactions in shared human-hippo landscapes. And but yeah, we'll have to stay tuned. We'll be working on this for the next couple of years. So if you have any questions or you're interested in this, please feel free to chat to me afterwards. Thank you.
Great, thank you so much. What a nice talk to finish off with. Thanks very much. Very interesting and lots of useful applications. Okay, hang on. Before you all leave, I've got 2 announcements, really important. As you know, we do work with community groups as part of our benefit sharing programs. There's one group of ladies outside that made the conference bags. They're selling nice little, very cute little car dustbins that you can use in your car, and a couple of handbags and things. We also work with another group that makes these laptop bags. Very nice, very, very nice. These are made with batik, so they actually make the fabric. And there's one available at the moment. So if anyone's interested, I can also make this available to you, and it's in support of some community sewing groups. So we're going to close off for the day. I think before we close, let's just give our sound and problem-solving chaps a round of applause. And then thanks very much to all the speakers for today, and we look forward to seeing you at the Cheese and Wine and at the dinner later today. And the game drive.